Hey guys, so today we are going to talk about the chromosomal theory of inheritance. And this is where genetics gets a little bit more complicated. So when we talk about genetics and genes acting as functional units as chromosomes or in chromosomes, they act a little bit differently than if we consider genes all by themselves. So we're going to be talking about the chromosomal theory of inheritance today and how genes react and how genetics work when we talk about chromosomes. So here we go. So the first thing I want to talk to you guys about today is pedigrees. We've got to learn how to read these pedigree charts, okay? So this is a pedigree, and a pedigree is a document that is just used to record ancestry. Oh, I know, I'm like, I can't get my board to work. What's going on with my board? I don't have workspace open. Hold tight. Technical difficulties. Okay. So a pedigree is a document that we use to record ancestry, and we use it by genealogists to study human family lines and stuff, but this definitely has genetics ties. So you can look at these pedigrees and say, oh, look, these parents didn't have the trait, or the grandparents or whatever, but these people have the trait. And it seems like only males are affected by this trait. So you can look at this and kind of decide, well, okay, well, what kind of trait are we looking at? So this is going to be our how to read pedigrees section, okay? So the first thing we need to know about pedigrees is that the squares represent males and the circles represent females. A shaded square or circle represents an affected individual. So this individual is affected by the trait. Now that could be a good affected, like it's a good trait, like I have an extra arm of awesomeness, or it's a bad trait, like it's a genetic disorder. So it doesn't really comment on whether these are good traits or bad traits, but on whether or not they have it or not. So yes, have it. No, don't have it. So unshaded is unaffected. If they're connected by horizontal lines, that means they're mates. Okay, so we're connected by a horizontal line. That meant we mated together. Doesn't necessarily mean they're married. This is not a legal document of any kind, but they are biologically mates. They had these girls as children, and these two had these children. All right. And if you're connected by a vertical line, that means you are parents and offspring. So here's a vertical line. That means here's the parents and here's the offspring. Vertical line, parents, offspring. Okay? Yeah, and you might be able to tell some weird fishiness going on in this random pedigree. But yeah, if you look right here, we have cousins that are mating with each other. That's a little bit weird. All right. So let's look at these pedigree charts, and I'm going to teach you kind of like the pedigree charts to know and what to make sure you're looking for when we look at pedigree charts. So this pedigree is a pedigree of an autosomal recessive trait. Now an autosome, everybody please say autosome, is a non-sex chromosome. So this is a trait that is inherited on a chromosome, and it's just not a sex chromosome. So it's one of the other 22 types of chromosomes that we have. So here's some clues that you're going to want to look at if you're looking at a pedigree to see, okay, is this autosomal recessive? So here's things to look at when we're looking for autosomal recessive traits. First off, males and females will be equally affected. That has to do with the fact that it's an autosome and not a sex chromosome. But here's the big deal. Unaffected parents will have affected children, and that is the biggest clue that we're looking at a recessive trait and not a dominant trait. So we look right here. Here are unaffected parents, so they don't have the trait, but they have affected children. And so that just says, oh, okay, so if the trait is like big A, little a, then these kids must be little a, right? Because they must have these parents don't have the trait, right? So they must have a big letter. But since their kids have the traits, they have to be little a. So they must have gotten a little a from dad and a little a from mom, okay? The trait skips generations, and we end up with carriers. So everybody please say carrier. A carrier is a person who doesn't have the trait, like these parents. They're carriers. They don't have the trait, but they can pass it on to their offspring. So they're heterozygous. They're big A, little a, all right? So remember, this is a recessive trait, so it only shows up if we're little a in this case. All right. Next up, a trait or a pedigree to make sure you're able to identify is any autosomal dominant pedigrees. So autosomal, again, is not a sex chromosome, so that means that males and females will be equally affected. Okay. And then affected children have affected parents. So this affected children has affected parents. This affected child has affected parents. You're not going to have any surprises. The trait's going to make sure to show up in each generation. All right, and that's just a dominant inheritance pattern. So these guys are big A something, big A something. It means this kid is big A something, this kid is big A something. But that means that these kids have to be little a, right? Because they don't have the trait. It's a dominant trait. So if they had even one big A, they would have it. So we can use that to figure out, well, what must the parents be? Well, since they have affected children, they must have gotten their big A's. But since we have unaffected children, that means that the parents must have been little a or big A, little a, so that they could get little A's from both parents. Right? So those are patterns to look for. And then you got these children that will not have affected children because it's not in their genes. They don't have that dominant trait. So here you have the potential to escape whatever that trait is by breeding. All right. So kind of a question, looking at this pedigree, would you say this is a dominant or a recessive trait that we're looking at here? So you'll notice that males and females seem to be equally affected, so it's going to be definitely autosomal. It shows up in every generation, and that's your biggest clue that that is a dominant trait. Okay, what about this one? Dominant or recessive? Yeah, we definitely have a recessive trait because it's like, oh, look, the trait all of a sudden just pops up out of nowhere when your parents didn't have it. Okay, so which individuals in this pedigree must be carriers? Who must have passed it on, right? Who must carry the bad gene? So let's look. 1-1 and 1-3. So it's asking, must they be carriers? Well, I don't know. This person must have gotten the gene from one of these guys, but I don't know if the carriers are the dads or the moms. So I'm going to hold off on that. 1-1 and 1-2. So they're saying that these guys are the carriers. Well, once again, I don't know if these guys are the carriers or if these guys are the carriers. So I just don't know. So 1-3 and 1-4, the same problem. Okay, 2-1 and 2-2. Yeah, they have to be carriers because they do give that to their children. So I don't know where they got it from, which parents they got it from. Probably one on each side, honestly. So somebody must be a carrier here and somebody must be a carrier here, but that's all I can tell. Okay, so then there's some more rare pedigrees that we might find. So this one is a pedigree of a mitochondrial disorder. All right. And so a mitochondrial disorder is a disorder where the mitochondria doesn't work, and that's a big problem. Mitochondria disorders are fairly deadly. So here's the deal, though. Mitochondria are only passed on by women, by the eggs. And so a male cannot pass on mitochondrial damage. They don't pass on their mitochondrial DNA. Since the eggs are what holds the mitochondria, and the sperm, they just hold DNA because they're lame. Not really, they're not lame. I didn't say that. Only mom passes on the mitochondrial DNA. Dad does not pass on the mitochondrial DNA. Okay, so here's clues about what you might look for in a mitochondrial pedigree. It shows up every generation, so every generation will have it. There's not a diploid condition. It is you have it or you don't have it. It's not like big A, little a, or anything like that. There's no dominant recessive. It's, do you have it from mom's egg or do you not have it from mom's egg? Okay. And it's maternal transmission only. That is the biggest thing. And also, all offspring will express that trait. So if mom has it, everybody's got it. Okay? So here, mom has it, everybody's got it. Mom has it, everybody's got it. Dad has it, nobody's got it because mom has to be the one to pass that on. So it's a weird one. So if you're like trying to figure it out, man, that is a weird pedigree. It looks maybe dominant, but only mom's passing it on. You might think, oh, maybe it's mitochondrial. All right, we're going to come back to those pedigrees in a minute, but we've got to talk first about sex-linked traits. So everybody please say sex-linked traits. Cool. Sex-linked traits are genes carried on sex chromosomes. So we've been spending time talking about autosomes. Let's talk about your sex chromosomes. So gender in humans is actually initially determined, and we could have gender sex debates, and we could, but I don't want to right now because you're not here and I'm alone. That's determined by the Y chromosome. And honestly, it's not much of a debate. In Mrs. Funk's opinion, you can pick this. This is a little bit more fluidy. I don't know. I'm very open here. Whatever you want to be you. Anyway, Move on, Funk. Move on quickly. So the human X chromosome is one of our chromosomes. It has more than one of our sex chromosomes. It has more than 1,000 protein encoding genes. That means more than 1,000 useful genes. Okay, and most have nothing to do with sex determination. So your X chromosome is a very important chromosome. It's needed by males and females. So you have to have one. A female is XX. All right, so that gives you a female genotype, XX. A male is XY. That would give you the male genotype and phenotype. Um, and so you can tell everybody has to have an X chromosome. They're very important chromosomes. 1,000 genes, that's a lot out of our 20,000. That's a lot. Okay, and most have nothing to do with sex. That's why both females and males have to have them, because there are keep you alive kind of genes. Okay, if you don't have one, you're dead. You're not even around. Okay, the human Y chromosome though has that large role in sex determination, and it only has about 100 genes on it. So females, you don't get the Y chromosome. That's why you're female. Males, you get the Y chromosome. That's why you're male. All right. Those chromosomes turn on at some point during, or those genes turn on at some point during development if you have one, and give you the male anatomy. If you don't have one, those genes obviously wouldn't turn on and wouldn't give you the male anatomy. All right. So, and then this is how it's determined. You get an X from mom's eggs, because that's all mom can give you. Dad could give you an X or a Y during meiosis. This is diploid, right? And they split up. So some of dad's sperm will have X's, some of dad's sperm will have Y's. And then if this sperm happens to fertilize this egg, it's XX. If this sperm happens to fertilize this egg, it's XX. If this sperm happens to fertilize this egg, it's XY. And if this sperm happens to fertilize that egg, it's XY. Cool. All right. So next we have X-linked recessive color blindness. All right. So this is a trait that is present on X chromosomes, some X chromosomes. It's a gene that can go bad. All right. And so we say that it is X-linked because it's a gene that we find on the X chromosome. I have no idea where it is. Maybe this is the bad part. I don't know. All right. So here's my X chromosome. It's really big. Here's my little tiny Y chromosome. It's really small. Okay. So this is like a trait that's on, it's a recessive trait that's carried by the X chromosome. It's linked to the X chromosome. That just means it's on the X chromosome. So this is a carrier mother. She's X big A, X little a. So she's normal, but she's a carrier for color blindness. And then here's the father who's normal. And mother can, since she's a carrier, she can pass that on to her children. Now, what's really interesting with X-linked traits is that males are much more likely to get these traits than females. And it's because males only have one X chromosome. Females have two X chromosomes. So a male, he kind of only gets one shot at a good chromosome. And if so, he could have gotten mom's good chromosome, or he could have gotten mom's bad chromosome. And since he just happened to get mom's bad chromosome, he's colorblind. But females, they have, as long as it's a recessive trait, they have double the chance of getting a good chromosome. So this female, she has a normal chromosome and an affected chromosome. Since this is dominant, she's not affected with color blindness. She's also a carrier though, so she could pass it on to her kids, but she's normal. So it's really interesting with these traits. You're going to see more males getting sex-linked traits than females getting sex-linked traits. Highlight. So this is color blindness. I guess I should have talked about that first. People with color blindness would not be able to distinguish the colors in these circles and would not be able to see the green number 7 sitting there. So if you can't see a green number 7, potentially you could have red-green color blindness. Red-green color blindness affects around 8% of males in the United States. That is an X-linked recessive color blindness. There's another color blindness. It's a blue color blindness, but that is an autosome, so it's not sex-linked. So males and females are equally likely to get it, and it's dominant. So that one's kind of interesting. So is it possible to have a female? Oh, hello. Sorry, I'm recording.
No, you're fine. You're sucking out the other person.
Sorry, guys. I found out that I'm going to have to take a break anyway because they're going to come clean my sinks. But let's do as much as we can before I have to do that. So much to do today. So is it possible to have a female that is red-green colorblind? Of course it is. She would just have to have X little a, X little a. So her mom would have to give it to her and her dad would have to give it to her. So that tells you the only way to have a female who's colorblind is to have a dad that's colorblind. Okay, so let's look at how these sex-linked traits or these X-linked traits are transmitted, okay? So here you have an affected male, so he's affected, he's colorblind or whatever, and a homozygous dominant female. So if we do that Punnett square, here's the affected male and here's the female. This sperm fertilizes this egg, that's a female, but she's a carrier. This sperm fertilizes this egg, that's a female, but she's a carrier. All the boys are normal because mom only had normal X's. So in this Punnett square, all the males are normal and all the females are carriers because dad had to give them that affected trait. So if dad's colorblind, all of his daughters will at least be carriers. And if mom happened to be a carrier, some of her daughters might be colorblind. So here's this Punnett square. Here's a normal male with a carrier female. We saw this one. So you've got half of your female children will be carriers because X big A, X big A, X big A, X little a. Half of your male children will be affected because X big A Y, that's a normal son, and then X little a Y, that's an affected male. Okay, you can also have affected females and or carrier females and affected males. That would be the only way to get an affected female daughter because dad would have to give you a bad copy and mom would have to give you a bad copy and dad would have to give you a bad copy. All right, the males will be one of the males will be affected because mom's a carrier, and then about a fourth of the males won't be affected. All the daughters are either a carrier or they're affected, though. That's cool. That's cool. All right, so this is a pedigree for sex-linked recessive traits and stuff to make sure that you're looking, and what to make sure you're looking for when you think you have sex-linked pedigree. So clues about whether you're looking at an X-linked recessive trait. Definitely this one. If you see many more males affected than females, okay, you might start thinking, oh, maybe it's sex-linked. If you see an affected son with normal parents, okay, so here's an affected son with normal parents, and any affected females must have an affected father, okay, and a carrier mother or an affected mother. So the mother can be a carrier or affected. So females must have, oh, it's not showing her dad, must have an affected dad, and mom must have been a carrier in order to get females. So they're going to be really rare females that have it. So this is a pedigree of Queen Victoria, and this is not for color blindness, unfortunately. This is for hemophilia, which is a disorder where blood does not clot correctly, and pretty much you bleed to death from fairly simple things. So we think that the trait actually started with Queen Victoria and that she was a carrier, at least of this line of hemophilia. And who knows where she got it from or if she originated the mutation. I'm not sure. I bet you could look that up. But so she was a carrier. So she was X big A, X little a for hemophilia. And so none of her daughters had hemophilia because their father didn't have it, but she did have multiple carrier daughters, and that is going to come back later. And she had a son, Leopold. He died at a fairly young age of hemophilia. Okay, he did have children who were one of which was a carrier, and then they had a son, Rupert, later on that died of hemophilia too. But what's really interesting, since royal families like to marry other royal families, right? Queen Victoria's daughters all married into different royal families. So you've got the modern English family, you've got the Spanish and German and Russian royal family. And since Queen Victoria's daughters married into each of these families, hemophilia is actually a problem in the European royal families. It's just been passed down. And yeah, it is a problem that primarily affects the males of that, and the females are just carriers. So looking at this pedigree, what do you think we're looking at? Okay, is it autosomal dominant, autosomal recessive, X-linked, or mitochondrial? So first off, we can let's get mitochondrial off the list because if you look, there's an affected mother, and not all of her children are affected. So so this is definitely not mitochondrial. Let's try dominant. It could potentially be dominant because you look, it shows up in every generation. It looks like males and females are affected equally. So in my mind, this seems dominant to me. It doesn't seem recessive because it shows up in every generation. It's not skipping. Let's look at sex linked, if it could be sex linked. So sex linked, we would have a male with an affected mother, maybe. I don't know about this one though. It just seems like there's the same amount of females and males being affected. So I'm going to look at this one. So here's a female. She would have to have an affected mom or a carrier or an affected dad and affected. So that kind of works. Yeah. Yeah. It was what I thought it was. It's dominant, and it's just that showing up, and it's equally affected. Okay. So do you guys think that you have to have two chromosomes? And of course not. If males don't need two X chromosomes to survive, females don't need two X chromosomes to survive either. So actually, females, since you have two X chromosomes, in your cells will choose to deactivate one of those X chromosomes and create something called a bar body. Okay, so that is a deactivated X chromosome. So this is called X inactivation, and it's where one of the X chromosomes is turned off early in female development. And so your cells will say, oh, we're just going to turn that off. Now, what's really interesting is cells don't necessarily talk to each other about which X chromosome they're going to turn off. So if this is the ovum and this is a female, she got one X from mom, one X from dad, our cells are going to start dividing and during mitosis to create her. And right in here, after there's early cell division, so this is after mom's an embryo, which is basically a ball of a few million, I don't even actually know how many cells, it might even just be hundreds, a ball of just a few hundred cells. Some of them will decide to turn off dad's X chromosome, and some of them will decide to turn off mom's X chromosome. And it's just random. There's no talking. They don't go, we're going to all turn off mom's X chromosome and you're going to turn off dad's X chromosome. It's just random. And so what you get is you'll get a patchwork of some cells that turned off mom's X chromosome and some cells that turned off dad's X chromosome. And that can kind of create a weird mosaic, because if there's a problem with one of these chromosomes, like it seems like dad's sex chromosome, or was it mom's sex chromosome did not produce sex glands. So there are not sex glands, sweat glands. So this sex chromosome has a problem with the sex glands. And so half of this woman's cells are going to have a problem with the sweat glands, and half of her cells are not going to have a problem with the sweat glands. Those will be normal cells. I think I got that mixed up, but it doesn't matter. Okay, and this is called mosaicism, where females, they can be a mosaic of different traits even within their same body because of their inactivated sex chromosomes, which one got inactivated. And so they can have mom's traits in part of their body and dad's traits in the other part of their body. That's pretty dang cool. And this is how we get female calico cats, by the way. Calico cats are pretty interesting. So color is sex linked on cat. So on the sex chromosome, you can have orange allele or you can have a black allele. So if you're a male, you only get one. So a male cat can either be orange or a male cat can be black. And they can have white with that too. White is a different gene altogether. So white would be a different chromosome altogether. So they can still have white color. But a male cat, unless there's a mistake, cannot have both orange and black colors. But a female cat also, like, it's not, one cell's not going to be producing both orange and black. But what happens is early in the development, some of those cats will deactivate that chromosome, and some of the cat's cells will activate that chromosome, and you'll end up with patches, because you'll have a patch that's expressing mom's orange DNA, and you'll have a patch that's expressing dad's black DNA, which is really cool. And that's how you can have calico cats, is they'll be producing their own white that's on a different chromosome, their own white color. But then half of their cells will be producing black, and around half of their cells will be producing orange, and that will end up with the calico coloring. So Mendel contributed a lot to our understanding of genetics, right? He contributed that heritable material exists as discrete physical units. So he came up with the concept of the gene. And he said that these factors, these genes, are separated when we form the gametes. Like, he came up with the whole independent assortment and stuff, like without even knowing what meiosis is. As microscopes improved, we began looking at meiosis because Mendel never saw meiosis, and we saw, oh, those chromosomes, they behave like those things that Mendel was talking about. And that gave us our chromosomal theory of inheritance, that those genes are located on chromosomes, and the behavior of those genes is explained by meiosis. Okay, so let's talk really quick. We've talked about Thomas Hunt Morgan for a little bit. I want to talk about him a little bit more. So Thomas Hunt Morgan was really interested in gene linkage, and he came up with the first evidence that tied specific genes to specific chromosomes. So he says, look, these genes go on certain chromosomes. They're linked to certain chromosomes. And he reasoned that eye color must be an X-linked trait, because he noticed that when he was breeding, it was mostly males that were getting white eyes. Females usually had red eyes, and if there was a white gene trait in the population, it tended to be the males that had the white eyes. And he thought, well, that's really weird. Why would that not be equal? And so he said, okay, well, eye color must be sex linked on fruit flies. And that's what he figured out. So his next big breakthrough had to do with recombination, though. So he was figuring out that a high number of his offspring were normal. They had the parental phenotype due to the linked genes, due to the fact that they are on the same chromosome, so they do not assort independently. That means they're linked together, right? So mom's phenotype was red eyes, gray body. Dad's phenotype was red eyes. Let me look at the actual trait we're looking at. We're not looking at eyes here. That's what the issue is. Okay, so mom had gray body, normal wings. Dad had black body, vestigial wings. Okay, and he said, okay, these are on the same chromosome. So body color and wing type are on the same chromosome. Body color and wing type are on the same chromosome. And he says, since they're linked, these can't assort independently. These guys are going that direction. These guys are going that direction during meiosis. So he was finding that a lot of the babies had the parental phenotypes. And look at these numbers. 965 and 944 had the parental phenotypes, the gray normal and the black vestigial, right? They didn't separate. They stayed together, those traits, which was weird to him. But Hunt also discovered that there were some recombinant offspring that were explained by crossing over. The recombinant offspring, you'll notice they're a lot fewer in number, though, because it's 206 and 185 compared to almost 1,000 each of the other ones. So there's a lot fewer of the recombinants. But the recombinants, there must have been a crossover where these guys, they got mom's gray body, but they got dad's, must have crossed over, short wings. And these guys got dad's black body, but it must have crossed over because they got mom's long wings. So there was a crossing over event, a recombination. And so he figured out how to calculate a recombination frequency, which is how often genes are able to assort independently. Okay? And so linked genes rarely assort independently, right? Because they're linked on the same chromosome. The only way they would assort independently is if there was a crossing over. And crossing over is rare. So linked genes will have a recombination frequency of less than 50%, because they're going to assort independently less than 50% of the time. Genes that aren't linked, they cross over more than 50% of the time, or at least 50% of the time they cross over. So when genes are located on different chromosomes, they have a recombination frequency of 50% because they recombine half the time. They assort independently. All right, so that was really confusing, but you'll get there, I promise, okay? So which number of genes do you think would have a higher recombination frequency? So assort independently more often or crossover. Do you think AB is going to cross over more often? Or do you think YZ is going to cross over more often? And we've actually brought this up before. Which pairs of genes are more likely to cross over? And so we learned before that the farther apart genes are on a chromosome, the more likely a crossover, all right, will occur between them. So those genes A and B are not likely to cross over very often. They'll have a really low recombination frequency. But like A and Y, since they're further apart, they'll have a higher recombination frequency. So we'll use this. And we can use the percentage of the recombinant offspring to determine the distance, which is really cool. So we use the recombination percentage to determine the distance between two genes. I think this is the neatest thing. Okay, so if we look, these are linked genes, okay, A gene and B gene, A, B gene, all right? And it's saying if they're further apart, there's more likely crossover event. There'll be crossover events. But if there's closer together, there's less likely that there'll be crossover events. So here we have the gray normal. So we'll call gray normal for this parent. And this parent has, this is the same chromosome, black vestigial. And we want to decide, well, how far away are these genes? Okay, so what we can do is we can calculate the recombination frequency. It's pretty simple. Recombination frequency is just the, I thought I had a actual formula, but what? Apparently I don't. So it's going to be the recombinant divided by the total times 100%, and that'll give you your recombination frequency. Okay, so here's my number of flies. There's a total of 2,300 flies. Okay, and there are 391 recombinants, because if I added these together, those are my recombinants. There's 391 of them. So that's 0.17 or 17%. So I would say that these guys are 17 map units apart. So that's pretty interesting, and we'll look at recombination frequency again later. So how far apart do they have to be before they assort independently? 50 map units is the maximum value. If things are 50 map units apart, that means they recombine 50% of the time, and that's just like acting on separate chromosomes. So that's kind of the maximum. You really can't be any further than 50 map units apart, because at that point you cross over often enough that it's almost like independent assortment. Okay, so you'd be like 500 recombinants out of 1,000 flies. Come on, that's 50%. Those guys are as far apart as they get. Okay, I know that's weird. So when chromosomes assort independently, I have half a chance of getting a big A and half a chance of getting a little a, just like if they're 50 map units apart. Okay, all right, so we can use this to build maps. Okay, so if we look, I have gray body, normal wings, and wingless. There's a recombination. I'm going to actually start drawing this. Okay, so if I look, I'll just put B here, just kind of as a reference, and look at which is further apart, B to N, A to N, B to A. So it tells me A and B are pretty close together. N is all the way somewhere over here. So let's kind of like look at this. So there needs to be 18 map units between B and N. There needs to be 12 map units between A and N, and that puts 6 between A and B. So you kind of get practice doing that kind of thing. So let's look at this one. Yay. Okay. Determine the order of genes L, M, N, and O based on the following questions. Okay. So O and L are 45 map units apart. O and M are 35 map units apart. L and N are 20 map units apart, and M and N are 30 map units apart. So let's start with the furthest ones away, because that kind of helps us. So O and L are 45, so we know that this is 45 units. Okay? Now let's kind of take this, because now we've got to decide, well, where do all the rest of these guys fit in? Let's try O versus M is 35. M to N is 30, so I can't quite get there yet. So I think I'm going to go ahead. I'm going to put M somewhere over here, and I can erase this, okay? And that would be 35, which puts this at 10. 10 plus 35 is 45, okay? And now I need to find where N goes. Okay, L and N are 20 map units apart, so I could put N here. M and N are 30 map units apart. Oh my gosh. So I can't put it there. See how crazy this is? Does it give me an O and L? O and L, that was 45. Huh. I'm really bad at this kind of thinking, actually. I'll be really honest and upfront. This kind of thinking is so difficult for me. Okay. So L and N are 20 map units apart. So maybe I'll start there and do N is 20. Okay, and let's kind of check the map. M and N need to be 30, so that's not going to work. Okay, so what if, so this is 10. What if M is over here? And so this would be, so that lets O and L, and O and M are 35. No, that doesn't work either. O and M are only 35. Oh my gosh. Okay, so what if I put, this is M, because O and M are 35 map units away. O and L are 45 map units away. Okay, that makes this 10. So I'm still stuck on where to put N. Golly. Maybe it's L that's in the wrong place. No, because O and L are 45. Okay, so what if I put N over here? Let's try putting N here. So that would put L and N, there's 20. And then M and N, there's 30. So 10 plus 20 is 30. What about an O and N? Is there an O and N? There's not. And that would be over 55, which is fine. Just means they assort independently, basically. But I think we're there. So let's look. O-M-L-N. Let's see what we got. Yay! Oh, that was difficult. Okay, now sometimes we can have problems with chromosomes dividing, okay? And this is called a non-disjunction. So everybody please say non-disjunction. Okay, this is where the homologous chromosomes do not separate properly during meiosis. So here, where mom and dad's DNA is supposed to separate, there's a non-disjunction, and they don't separate. So they're stuck there. So when these guys split, this guy ends up with an extra set of chromosomes. And this guy is missing a set of chromosomes. So these guys are all going to have some issues. Whereas here we have another meiosis. This one goes just fine, but meiosis 2 has a non-disjunction. So here we have normal gametes, and here we have some abnormal gametes, some with too much DNA and some with not enough DNA. Okay, so there's two ways we can get non-disjunctions. We can have a non-disjunction during meiosis 1, or we can have a non-disjunction during meiosis 2, and it affects your gametes that way. Okay, so you can have aneuploidy. Everybody please say aneuploidy. Aneuploidy is where you have one extra chromosome. This is aneuploidy, aneuploidy, aneuploidy. Okay, so those are all forms of aneuploidy. And then you can have polyploidy. So everybody please say polyploidy. Polyploidy is not really pictured here. That would be an entire extra set of chromosomes. So that would be like triploid or quadruploid, tetraploid. That's the word, tetraploid. And we'll talk about how the heck that happens. It's going to happen pretty much with just plants. If this happened with humans, we'd be dead. All right. But we'll talk about how that happens later on. So Down syndrome is trisomy 21, and that just means this person, it's an aneuploidy, because this person has one extra chromosome. So see if you can see which one. I mean, it's kind of there for you. So chromosome 21, you can see there's 3 extra there. That was because of a non-disjunction. So that would have been probably an issue right here, where it's all normal except for one little extra chromosome, and that's that one right there. And this affects a person and gives them Down syndrome. So there's some this is the last slide. Doesn't that make you so happy? There's also some other issues that we can have with chromosomes too. We can have deletions where we delete whole segments. You could tell that'd probably be pretty bad depending on the gene. If this is just some junk DNA, well, then it doesn't matter. If this is really important DNA, then this person might even be dead. If it's not so important, maybe this person has a genetic disorder of some kind. You can have a duplication where you copy extra sets. Same thing. You can still have lots of issues with extra DNA. It depends on the gene. If it's a gene where having extra is okay, then it's okay. If it's a gene where having extra is bad, then it's bad. Inversions are where we reverse segments of chromosomes. So one BCD, now it's DCB. That person's usually okay. It depends where that break happens and how that gets inverted and what genes are being affected. Usually this person is fine though, except during meiosis. This usually tends to cause non-disjunction problems during meiosis because mom and dad's chromosomes have a hard time finding each other. during meiosis because they're not quite the same. And then translocation is where the chromosomes just swap DNA from one chromosome to another. It's pretty crazy. Once again, this person is usually okay. It depends on how those breakages happened, but they have a lot of trouble during meiosis because these chromosomes are going to have a really hard time finding their partner when mom and dad's DNA is trying to match up together. So these are just some other problems we can have with chromosome structure. All right, that was a lot. Hopefully not too stressful. Oh, I got through it anyway. Oh, there's the lights again. So have fun. Let me know if you need anything. Thank you, guys. Let's see. Turn off.